Second lecture. Start with the retina, and we introduce the concept of lateral inhibition, which is the idea that in the retina we see that excitation of certain photoreceptors not only causes excitation of some neurons that those are connected to, but can actually cause inhibition of the response to nearby neighbors. So you get this kind of opposite effect. So if you activate in this of the neuron that is activated by 3, 4, and 5. So it's kind of a shutting up or reduction of response to nearby neighbors. And that's basically the idea of lateral inhibition. And this was first discovered in the horseshoe crab or the limulus crab, where scientists discovered that if you activate a certain photoreceptor, in this case A, you get a high degree of frequency of response. But if you activate nearby photoreceptors, so here we're activating both A and B, that actually produces a reduced response from the electrode that's connected to A. So there's this particular horizontal projection that connects these, and so activation of nearby neighbors actually produces a reduced response of the original neuron photoreceptor here, A. Of lateral inhibition, when we look in figures such as this one, so even though these gray bars are actually uniform, so they're equally gray across within a bar, there's the illusion that this looks a little bit lighter in this region, and this region over here on this side may look a little bit darker than the rest of this bar. And that is attributed to lateral inhibition. The idea being that because this is receiving more light, this actually produces a strong inhibitory response to nearby neurons that are responsive to this region over here. And so that actually produces some darkening. And on the flip side, because this is dark, it's not producing as much inhibition, and therefore this produces a little bit lighter than this region over here, where you're getting a lot of inhibition from these other regions. So this is getting relatively less inhibition. And similarly, simultaneous contrast, this square over here tends to look a bit lighter than this one, which looks darker. And the idea is that because this is dark over here, it's not producing as much inhibition over here as this one over here, because it's light, is producing a stronger response from the photoreceptors and it's producing inhibition to its neighbor. Human eye, lateral inhibition is largely an effect of the wiring of the different layers of the retina. So we talked about the photoreceptors that converge on the bipolars, and then the bipolars converge on the ganglion cells. And one thing to note is that you start off with a lot more photoreceptors. You have 100 million photoreceptors, and by the time those end up converging on the ganglion cells, you have a massive convergence, and you're down to about 1.2 million ganglion cells, so about 1% as many ganglions as there are photoreceptors. And the ganglions, of course, are the ones that project out the back of the eye and go to the brain. So what's happening here is there's a massive amount of compression that's taking place. The original sensed image contains a lot more information than the image that ends up making its way back of your eye and out to the brain. And we talked briefly about some reasons this might be the case. Part of it is potentially neural processing limitation of the brain itself. The brain can only handle so much information. But perhaps more importantly is the limitation that the information has to leave the eye. And so if we had to get all of those photoreceptors axons out the back of the eye, that would lead to a much larger blind spot. So what we have to do is converge that information before and compress that information before that information leaves the eye. So we watched a little video of ganglion cell response. So this was a cat ganglion cell response that was responding to a stimulus that we were watching. And so this was a little ball of light that was a spot of light that's being moved around the screen. And the things to notice from this, I'm sorry, I can't figure out how to show the video right now while recording, but as the spot moved around, there was a certain location which, when that spot was there, when that spot of light was there, it would make the cell, make the neuron fire a lot. And when it wasn't in that region, it wouldn't fire. And also, when there was a big spot of light, the ganglion cell wouldn't fire as much as when it was a small spot of light. And when it was a big spot of light with a hole in the middle without light, that made it really not respond at all. And this brought up the idea of receptive fields. And receptive fields are the location on the retina in terms of the photoreceptor array that drive the response of a specific neuron. So if you're talking about a ganglion cell, what we mean by a receptive field is those neurons, those photoreceptors that make the ganglion cell fire more or less. So you can think of a ganglion cell's receptive field as being the photoreceptors that it's in charge of monitoring. So if those photoreceptors get activated, get hit by light, and it makes it respond more or it makes it respond less, then we say that those photoreceptors are in the receptive field of that ganglion cell. And as we mentioned, ganglion cells can be big. Receptive fields can be large. They can be small. So they can have receptive field that's covering a large portion of the image in terms of the photoreceptors that that light falls upon. It could be a small part of the image, and they can overlap as well. So you could have a single ganglion cell that responds to this, all of these points in the image, and then a different one that responds to all of these points in the image. And something else to note is this center surround structure that we're going to mention in a moment. And so there's this donut-like structure to the receptive field, to the way that the ganglion cells respond. And we also noted that receptive field sizes tend to grow as you get further out into the periphery. So these are large receptive fields, these are small receptive fields, and as you see, there are many more large receptive fields as you get far out in the periphery. And smaller receptive fields, there are considerably fewer of them. So ganglion cells' receptive fields have this center surround structure. What that means is that there is a portion of the photoreceptor array. So this is I can think of this as a chunk of the matrix of all those photoreceptors. There's a chunk of them that a portion of that's going to make a ganglion cell fire more, and a surrounding area around those, again, also made up of photoreceptors, that's going to make that ganglion cell respond less. And this is an on-center center surround receptive field, and this is an off-center one. And so that simply means is the center of this thing making that ganglion cell fire more or making that ganglion cell fire less. And whatever the center does, the surround is going to do the opposite. And this has the effect of sort of canceling each other out. So if you have a small spot of light in the center of an on-center surround receptive field, it's going to make this ganglion cell fire a certain amount. If you increase it to the point that it's really filling up the center, it's going to fire maximally. But if you get a little bigger, then if that spot of light gets a little larger and heads out into the inhibitory surround, it's going to reduce the response. And then if you cover the whole thing, you're going to be down to a very low response because there's going to be a lot of inhibition that's canceling out the excitatory response that's hitting the center. So how do you build one of these? Well, this is where the bipolar cells come in. This is why we have probably the 3 layers, or at least a portion of the reason we have the 3 layers. So the photoreceptors get activated. Some of the photoreceptors are connected to a bipolar cell that does one thing. So the center one, the bipolar cell, does one thing, and the ones on the opposite, on the surround, do the opposite of that. And that's why they're called bipolars, just because they can do 2 different things. They can either take an excitatory response and invert it and turn it into an inhibitory response, or they could take an excitatory response and not invert it and keep it and send it as an excitatory response to the receiving ganglion cell. So this is a way of building a ganglion cell that has this kind of center surround receptive field. In this case, it's an off-center receptive field because this is an inhibitory response. If these photoreceptors get excited, this bipolar is inverting it and turning it into an inhibitory response in the center. And these guys on the outside and the surround are not inhibitory, so it's excitatory. So that center spot is going to cause an excitatory response, and then the surround is going to cause an inhibitory response, and these guys are going to cause an excitatory response. And so that's how you get the center surround structure. So what is the point of this center surround? And so what we mentioned was that what it's able to do is it's able to detect places in the image where there are differences, where there are changes in illumination. This turns out to be potentially the very important understanding of or concept of what the ganglion cells are doing. So if you think about this, not much going on here. It's black all the way through. It's fairly uniform. So this receptive field, if we think about it as a center surround receptive field, not much is going to be happening here. And what's going to happen though at the edge is you're going to see a sudden change in the characteristic relative to the other ganglion cells. So let's talk about this one example over here. So it's getting excitatory, it's getting lit up, and it's getting a lot of excitatory information because it's falling on its center, which is excitatory in this case. And it's getting quite a bit of inhibitory as well, but it's not getting as much inhibitory as this one here. So this, because this part's in dark, it's not getting as much inhibitory, and therefore this is going to produce this kind of peak of response over here relative to its neighbors. And so this ganglion cell is going to be particularly active, more active than all the other ganglion cells that are nearby, because it's close to an edge. And this one over here is also going to be different. It's going to produce a sort of peak of low negative response because it's getting, in general, it's getting not that much excitation in its center, but it's not getting that as much inhibition either, but it's getting this little extra inhibition because this is falling in the light, relative to this one, which is getting excitation, not much excitation or inhibition. This one's getting a little more inhibition, so it's going to have this drop. So both of these are going to be, in a certain sense, different than all their neighbors. And so these guys are going to send their signals to the brain and this will be able to tell us where the edge is. It's going to tell us that this is where something interesting is happening relative to all the other parts of the retina. And so this potentially could lead to some sort of compression. That what's happening is that the ganglion cells are sending information about where the edges are and where there's changes in light on the image that the photoreceptors are receiving. So why would you want to do that? Well, because where there's a change in light is likely that that's going to be the edge of an object. So this might be a clue to the brain that this is actually something important going on in the image. If you have a sky like this one where nothing's going on, that's not something the brain doesn't need to know about every single point here where nothing's going on. What it wants to know is where there is a change in illumination, because that's an indication, and so that's what you're seeing here, these kind of edge structures, and that's an indication that there's actually something happening. That change is an indication that there may be the border of an object there. And so this is one way to think about what the ganglion cells are doing, is compressing the image and throwing out all the redundancy, all the information that's not really important, where there's uniformity, and sending the information as to where there's a change in the image.